and you're watching Epistasis. I just can't figure out this homework problem for genetics. Of course you don't. Let your genius older sister look at it. Do you even know what epistasis is? Well, my teacher said it was about genes wearing masks, but I don't understand why genes have to be protected from other people. No, you idiot. Your genes aren't wearing face masks. They're interacting with each other, and sometimes one gene can mask the effect of the other. Oh. You still don't get it, do you? Let me explain it to you in an example. There are 6 modes of epistasis that relay the way that genes expressed phenotypically are happening genotypically. Let's look at single dominant epistasis.
Having a dominant allele at one locus may prevent you from expressing the phenotype of the dominant allele at a different locus. Say that your features appeared to everyone else in filters such as normal, black and white, or comic book. If your genetic makeup happened to have the dominant A allele at locus 1, then you could not move past the enzymatic checkpoint because it's an inhibitor. Then you would have the no-filter precursor over your appearance. However, if you have 2 recessive alleles instead of the dominant allele at locus 1, you could move to the intermediate black filter, and then if you met the enzymatic checkpoint for locus 2, you could also move into the end product filter, which is the comic book. Let's do a few examples to get familiar with this. So if we were to use the example of 2 heterozygous parents crossing, we would get these Punnett squares with the values of 3/4 to 1/4 ratio between the dominant and recessive alleles. So to find the offspring, we would calculate the probability of one crossing with the other, and you can see that if you cross the big A allele with the big B allele, regardless of what the second allele is, we could get 9/16 based on the ratios from the Punnett squares. So if we follow this out through all the other possibilities, we can get that we get a 9 to 3 to 3 to 1 ratio. And in analyzing the phenotypes, we can see that 12 would be normal filtered, 3 would be comic book filtered, and 1 would be black and white filtered. And these phenotypes express whether or not the dominant allele was present at the first or second locus and how the first locus affected the second locus. If we were to cross a different set of parents, we can see that the offspring might have different possibilities for what their phenotypes will be. So based on this, the genotype for the children might be big A, big B, or little a, big B. And we can see that this is actually an 8 to 8 ratio, which means that 8 would have normal filter and 8 would have comic book filter, and there would be no intermediate children.
You got that? Ah, I think I've got it now. Thanks, sis.